Well, good morning. Second time, second day. In another way, I have to continue pretty much the same talk, but with different aspects of my work. So yesterday we talked about the role of myosin A, 5A, in neurodegeneration. We tried to say that yes, myosin 5A is involved in etiology or pathogenesis of Parkinson's disease. But as you can see today, from the previous talk, there are so many other things, which can be also involved in etiology or pathogenesis of Parkinson's disease, like gangliosides, And so it doesn't really matter, It's complicate the matter more, because you figure out that there are so many factors which has something to do in one or another, with development of Parkinson or other neurodegenerative disorders, And so today what I'm going to do, is just try to continue what I started yesterday. Will be some overlapping for some people which were yesterday here, but I noticed that some people in audience, are different than yesterday, So for those of you, I will try to remind or try to present, the data which I had yesterday, but we'll see. So let's go with the next slide. Outline of my talk today is going to be a rat model, for neurodegenerative disease and the role of alpha-synuclein and tau in a BD4 rat model. BD4 means Berlin-Ducray rat, 4 rat model for neurodegeneration. And to tell you a little bit about the history of this BD4 or Berlin-Ducray rat, is coming from Germany, from here. Was developed during the Second World War by Ducray, and he developed 9 lines of BD4 rats, and every one of them, they have various sensitivity to chemical carcinogenesis, At that time, I was a graduate student at The Ohio State University, and my chairman, Dr. Kussner, he believed that, okay, we need to try to import some of those BD4 rats which are resistant to neuronal development of chemical-induced tumor of the nervous system. And one of those was Berlin-Ducray for rats, And we inoculate them with ethyl nitrosourea, to develop tumor of the nervous system, But instead, they did not develop that many tumor of the nervous system. They developed some schwannomas of the trigeminal nerve, but they developed other type of tumor, like breast cancer, ovarian cancer, and so I said that, well, these rats is okay, to study some other things, and I shift this my work, in chemical carcinogenesis of breast cancer and other things. And so I kept this strain of rats, until now. I moved them from Ohio State to Michigan State, and from Michigan State, in the back of my truck, I moved them to Texas, And so it's the only one which now we have in Texas. And I'll tell you the story of this one. Then we'll continue with genetic analysis and something about myosin 5A mutation, which we discussed already yesterday. I'm going to touch the subject only briefly today about this mutation. And finally, it will be the conclusion and future direction about these studies. As a rat model for neurodegenerative disease, I will have to say that this is not a transgenic rat. This is not a chemically induced model. Is just a spontaneous autosomal recessive model, And so that's probably what we need to develop. It's not because we develop them. They develop themselves, They come themselves, because that was the problem, When I tried to patent this type of rat model, of Texas A&M University, he said, well, you did not do anything, just did for themselves, And so I could not patent this rat model, It's mine. It was developed in my colony, but I did not create it, It was just coming, And the first creation of this one was in a Sprague-Dawley rats. At that time, I had a graduate student from Portugal, Odette Mendes, which worked in a breast cancer, When she worked in a breast cancer, she used this model, to induce tumor of the breast, and use another strain like Sprague-Dawley rats. Sprague-Dawley rats is good for those kind of things. And so finally, what she noticed in one day, that one of the mama developed 10 pups, and from those 10 pups, 3 of them are not doing well. About 10, 15 days, they start shaking, And she said, doctor, I believe that it's something wrong with the mamas, because apparently it doesn't have enough milk, to feed all of them. I said, I don't think so, Finally, we terminate them, and I suggested to her, please do the following, Try to breed the mama with the father, the same father, one more time to see what happened. And so what happened, repeat exactly the same situation, with 10 pups or 12 pups, 3 or 4 of them affected in exactly the same way. About 15 days, you recognized some movement disorder in those rats, and then situation become worse, and finally we had to terminate them about 35 days, because of the neurological defects, So we terminate them. And then I said, well, let's, it's outbred, it's not syngeneic rats. Let's move them, because I have the BD4 rats, which is a black hooded rat. Let's move them in a syngeneic rat, and we move them by breeding in the syngeneic rat BD4. And so in this way, I have now the BD4 rats colony with this mutation, starting from the Sprague-Dawley, but transferred into the BD4 rat, And of course, we didn't know the marker. We didn't have any genetic finding what's going on, and the breeding was done randomly, And so we tried to say, okay, let's choose this male, let's choose this female, see what happens, But that's very costly, because you spend a lot of money, and you get some results, but not exactly the way. It's not economical. And finally, we were able to detect, with the help of some geneticists from Baylor College of Medicine, that yes, there is a mutation, and this mutation was related to the myosin 5A, and we got a marker, and with genetic marker, we were able to restrain or reduce the number of colony because it's too costly. They cost me about random breeding about $2,000 a month, which is a lot of money. And I said, no, it's not good to have that kind. And then we transferred the phenotype, with the movement disorder in BD4 rats by inbreeding, okay? And then what I want to say about this model, is that they mimic exactly the same phenomena, clinical sign as it is in humans with Parkinson's disease, like tremors, spasticity, rigidity, bradykinesia, and postural instability. And for those of you which were yesterday here, you noticed that I showed you that movie, how they move. Today I'm probably not going the same movie because there are only 2 or 3 people which were not yesterday here. And so, as a neuropathologist, this model, what I try to tell you today is going to be about related to phenotype identity, increased alpha-synuclein and tau expression, neuronal degeneration in a specific brain area, Lewy body formation, neurons and neurites, and also inflammation, oxidative stress, mitochondrial dysfunction, and neurotrophic factor alteration, dopamine metabolic alteration. Those 2 ones about metabolic alteration and neurotrophic factor alteration were discussed yesterday, and so today I'm going to touch this one only briefly. So here they are, in the first breeding, when we transfer them, the first generation, we transfer them, from the Sprague-Dawley into the BD4 rats. You see a mixture of them, you see white here, and then you see those ones which are black, hooded rats, it's a real BD4 rats, and then you see the affected one in color. That draw my attention, I said changing color, why the affected one, they have that gray color, but doesn't have the black color like the other one, And so I said, must be something related to that. At 15 days, I noticed that those ones which have gray color of the hair are indeed the ones that carry mutation, myosin 5A, And we'll develop the story a little bit later in the next slide. And you can see in the next slide, when already established this type of colony here, it's a really clear-cut gray color in affected one, and black hooded rat, which is normal, move around, no modification in the movement disorder in this rat here. And so the first alteration, which we noticed was simply, take the hair, look in the hair, and you see these blobs of melanin accumulation in the hair shaft over there. And that is exactly what was described in the literature, in Griscelli syndrome, Eliade syndrome, and in other type of models, like mouse model for dilute syndrome, opisthotonus in a rat model by a Japanese group, with the problem changing color of the hair. And that is because of the lack of myosin 5A or mutation in 5A over there is not moving, the melanosome inside of the hair shaft to deposit the melanin inside of the keratinocytes of the hair. And you can see over there, some of the hair doesn't have anything in it. You can see here, some blobs accumulate here because the myosin 5A is not able to move the melanin to the target cells. Then the first pathology, I said, well, let's explore. Is anything else? Because the story was that no neurological pathology exists, besides the fact that it's some related to the hair. The Japanese group was able to detect, something related to the Purkinje cell spine. They noticed that the endoplasmic reticulum is missing in the spine of the dendrites of the Purkinje cell. And the conclusion for them was that, well, probably that's the way it is. They don't move because they have this problem, in the Purkinje cell dendrites over there. They do not move properly. And why they die, they say, well, probably because the mama doesn't feed them properly, But the problem is you can keep them alive, later, up to 15 days if you feed them, give them some supplementation if they cannot reach mama, because when you have 10 or 15 pups in a cage over there, those affected one, will be pretty much overwhelmed by the other one. It's a competition for milk, And then the other things you have to keep in mind, that as long as they suck the milk from the mama, they receive a lot of good factors like BDNF and other factors which help them to continue growth and continue to develop normally. After that, you have to supplement them with something and you can keep them alive for a longer period of time. And the lesion which draw attention, that are other pathology here related with the myosin 5A mutation, was the change in the olfactory bulb. As you can see here, the affected area was practically the mitral cells, and degeneration. And here it's a good example, as I showed yesterday here, of increased eosinophilia in the neurons, axonal neurons, from the striatum and also from the frontal cortex in the same time, which indicate to me as a neuropathologist that most likely the dying of these neurons is happening backwards, it's coming from the postsynaptic side, is going upward toward the center. And finally, you can see the death of the neurons, but later on, first the axon will be gone, and then will be the neuron's nucleus. The next one, we'll try to see if confirm the presence of Lewy body in these models, which hematoxylin and eosin you can see over here are some inclusion of pericardia with using specific antibody for the alpha-synuclein. We see also accumulation here in the substantia nigra neurons here, and another one is here, and also plenty of staining, positive staining here for alpha-synuclein and the substantia nigra neurons here. And so it fits the pattern what is described in the human literature related to the Parkinson's disease, the Lewy bodies. Next one, we're also using the same type of antibody, but in this case phosphorylated type, because of wild type, phosphorylated and nitrosylated type of antibody you can use. And in this case, you can see with specific antibody here in the striatum here, the presence of Lewy body, and here is a section also from the striatum, show the pericardia positive, for this accumulation of alpha-synuclein. This is the control here, and this is the substantia nigra, stained with nitrosylated alpha-synuclein. You see strong positivity here in this area here. And this is a larger area to show accumulation of those substantia nigra into the neurites here, where the arrow is there. And enlarged here also you see a pericardia cytoplasm for that connection a lot of punctiform accumulation of alpha-synuclein over there at the synaptic level. Then the next one, with this magnification, I know how good it's going to show because of the lights, but it's showing a control here in the striatum here. The positivity should show a punctate type of pattern here throughout the section over there, but not very clear. I would say the alpha-synuclein, you have to take it for granted. It's a very good staining for synaptic. You can pinpoint if a good technique is used over there with alpha-synuclein antibody. You can really pinpoint exactly where are the synapses in the nervous system, central nervous system. And here is another section with the phosphorylated type in the substantia nigra here. Phosphorylated type, you saw it here. And show it here, two pictures here, a different time point, early time point, later time point, about 35 days. Strong positivity in the substantia nigra with alpha-synuclein. The next one, it's exactly what we look for. And you can see here with the Western blot, a decrease in affected one of tyrosine hydroxylase. And this is a normal appearance histological section of the substantia nigra. Looked like a mustache of the last emperor of the Austro-Hungarian Empire, Franz Joseph, the mustache like that. You can see distinguished portion of the substantia nigra here, pars compacta, pars reticulata, and ventral tegmental area here in the normal one. And here in the affected one, you see some loss, We did some stereology, to look for the loss of neurons. And I have to tell you straight that the loss of neurons does not occur statistically significant in this case. It has some degree of loss of substantia nigra neurons, but it's not that severe. The most affected areas of the nervous system in this case, in this model, is related with the projections, of the axon into the striatum over there. And so the striatum has a lot of ramification in other parts of the brain, and that's the reason you see many other problems. In other things, we look at the alpha-synuclein, we look at the basal ganglia, some at the base of the heart, as you can see here. It's a positivity in those Lewis body-like small pinpoint over there. And another interesting one was the presence of alpha-synuclein in the heart, but only in the atrium, primarily staining the endothelial lining, as you can see here, endothelial lining here, but also in some cells, but very heavy infiltration of the endothelial cells with alpha-synuclein in this case. But not the ventricle, only the atrium. The ventricle didn't show any positivity, the myofiber for alpha-synuclein with this staining. Next one, yesterday I present to you this section here, but I not present you the other one section over there. The other one section over there, it's also interesting to look at, because it's related with the striatum over there. I use so-called Golgi cup technique, method of staining like this one, which is very, very laborious, very tedious, and takes a lot of time to do it right. It takes about a couple of months, you have to put in a specialty that you buy from over there. It's going to stay for a couple of months. Then you have to have a good histopathologist, which got the good section, but the good section has to be 100, 200 micron thick, And sometimes to keep the tissue together is very difficult, because otherwise they crack, they split, and it's very difficult to see any good picture. But if you have a good technician, yes, you will be able to get a good section. And so what I observe in this, with using this technique, give me a good clue, what does it mean to really look for the pathology of the nervous system in detail. And the pathology is going to tell you what's coming next. Then you have to look at the biochemical changes, you have to look at the genetic changes, and so you have to connect them. But the basic things for me as a neuropathologist is start with very basic, pathology to see exactly what happened over there. And if you use your mind, your imagination, then you can go to the next step, biochemical analysis, molecular biology studies, and then you go into the genetic to explain the phenomena. And so what happened in this case, you see the main neurons in a striatum over there are so-called spiny neurons. The spiny neurons are GABAergic neurons. They are about 95% of them populated area. They are GABAergic, and they have a lot of dendrites. In the dendrites, they have with high magnification here. You can see the dendrites here. They have a lot of spines. You recognize the space is very well. But in affected one, when they start dying, when you see here, the dendrites start losing their spines, you know. And then when you look here at high magnification, you see how few of them still in place and probably non-functional and show that kind of blobby-like appearance of the spine. You know. What it's telling me is telling me that these neurons not only are dying, you know, but are non-functional. And so this happens gradually. It's not happen only in the cerebellum, it happens probably in hippocampal formation. And that's the reason we lose our memory, you know, we lose our spine. So the memory is there in the spines. And they communicate through the spines. You know. It's like you see, and many times I make a similarity with the trees outside. You know. It's a good model to compare over there. You know. You see the tree, it's like the neurons, you know, with the deep roots going down. You know. You see the branches, those are the dendrites. You see the leaves are the spines, you know, to communicate with the universe, you know, to take what they need. This is a kind of philosophical approach to the problem, you know, that has something to do with the nervous system too. Then we look at the tau, you know, later on we look at the tau. When you look at the tau, you know, there's another component, you know, associated with neurodegeneration. Sometimes it's possible to see association with Parkinson's disease, but mostly with Alzheimer's disease or other neurodegenerative disorders, you know. And so I say, well, it's here. Why is here, you know? It's expressed in a brain stem here. You see some pericardial involvement here, positivity here, and also expressed in some specific area of the cerebellum. Those are nuclei in the cerebellum. Here you can see it's a pinpoint related to the synapses. But also it's expressed widely, you know, in the white matter more than in the gray matter, you know. And here is a section of the brainstem here. You show the strong positivity. The same kind of pattern we see in a striatum in some area later on, about 25, 30 days postnatal in this rat model. And so the tau is there. So are two components which are common find in neurodegeneration. One is alpha-synuclein, connect very well with Parkinson's disease. The other one is the tau. The tau associate more with Alzheimer's disease. Then we showed this yesterday, you know, and I just briefly touched the base here, what we did here, the expression of the tau in different parts of the brain, phosphorylated tau, you know, at different time point, 25 days, 30 days, and 25 days. I tried to check at 15 days, you know, early time point to see if there's any change in those components, you know, those proteins. No change, no difference between control and affected one. You see the changes after 15 days up, you know. So it's a time point, you know, when you see increase in time of severity of the disease and accumulation of these proteins here. The next one, we look at neuroinflammation. Neuroinflammation is also associated. We discussed yesterday. Many times I heard a lot of talks about neuroinflammation, a common problem in the neuroinflammation nervous system. Who are the components, you know, of inflammation in the nervous system? Based on what we were taught in school, you know, when I was in veterinary school, was that neuroinflammation is when you have some cells from the hematopoietic system, like neutrophil, macrophage, going over there, lymphocytes, and induce inflammation. It's not so. You have cells in the central nervous system, you know, like astrocytes and microglial cells, which can produce the same phenomena, you know, as it is with the hematopoietic cells, with some degree. And so what we found here, we found that there is a direct association between the increased number of astrocytes and microglial cells, significant, you know. And why they do over there? What did they do there? Is nothing wrong. It's no other inflammatory. You have no virus, you have no bacterial inflammation, but you still have the presence of those cells, astrocytes and microglial cells, in a large number, you know. Why do they do that? Good things or bad things? In a normal situation, those microglial cells, astrocytes, they do good things, you know. But this is an abnormal situation. They probably try to maintain somehow the homeostatic environment in a normal level, but they cannot do it because something else happened. Neurons are dying or neurons are affected, The proteins which are coming and interconnect, and make function properly with the astrocytic or glial cells are not helping them, or glia are not helping them. They release a lot of inflammatory cytokines, which can damage the neurons instead of helping neurons to grow. So that's the connection here. And you can see here, graphic representation here, the presence of IL-2 in a microglial cells, and also you can see this amoeboidal type of shape of the microglial cells. That's recognized very easily on immunohistochemistry. When you see that spread, like a bush, of microglial cells, they're activated. And also the astrocytes also show activation here. The next one we did also for GFAP to prove that indeed it's so. You can see here that there is increased GFAP presence in the Western blot, and also you can see activation of the astrocytes in various parts of the brain. So neuroinflammation is present over there. Are other things, related to this alpha-synuclein is not only this rat model or Parkinson's disease. The alpha-synucleinopathies, are common in Parkinson's disease, in multiple system atrophy, and also dementia with Lewy body formation. So I believe was somebody else to talk, Dr. Jenninger, I don't know if he came or not, to talk about multiple system atrophy here. If it's not, too bad, because I'm not going to talk about that one. This one we talked yesterday, because we did Western blots to show that it's indeed increasing alpha-synuclein in the brain. That was the first thing we see here. And then we saw that the alpha-synuclein, which is phosphorylated, is highly elevated compared with the other one in the brain of affected rats. We look at the messenger RNA, it's also increasing time point 20 days and 27 days in various parts of the brain, primarily in the mesencephalic area, where it's related to the substantia nigra in dopamine formation. So we go one step further here to look at the alpha-synuclein accumulation in the presynaptic terminals. Why I make the connection with alpha-synuclein and tau and myosin 5A? All three of them are located in the presynaptic side. Presynaptic side over there, sit there, doing nothing. The one which they move them out is the myosin 5A. The myosin 5A attached to those proteins, and move them postsynaptic, and everything will be fine. It's not only the myosin 5A attached with those two components, because there are 300 or more than 300 other secretory granules in the presynaptic side, neurotransmitters. If they are connected to this myosin 5A, let them move or not, I don't know. They have to be investigated step by step, but I believe probably that happened, And so practically we see that there is an accumulation of the alpha-synuclein in the presynaptic side. The other things we want to prove how many of these alpha-synuclein accumulate. It's a so-called technique, related with the section of the paraffin section. Then you treat them with proteinase K, and then you see the aggregates, insoluble form of the alpha-synuclein, and you can do immunohistochemistry exactly as you do with the slides from the tissue culture or the other slide. But you see only the ones which are not soluble, which are resistant to proteinase K digestion here. The next step is the look at the neuropathology, phenotyping, counting, as protein aggregation filtration technique, and you can see here again, it's an increase in olfactory bulb, mesencephalic area, and putamen in these rats, and mostly affected in time point at 15 days, 20 days, 25 days increase in this alpha-synuclein. Mitochondrial dysfunction. So I remember yesterday, Dr. Ogoyeva, also present the same thing in mitochondrial dysfunction. It's here too. The complex one is affected here. You can see mostly affecting the putamen, olfactory bulb, not that much, but putamen is affected, mesencephalic area is affected. Look at the complex one in this case. And then the next one, we did a confocal microscopy here to find out if this 5 rush because it's not enough to say. So the only thing I want to say is always time problem. Time problem is the thing you cannot say everything you want. The mitochondria is also in the associated alpha-synuclein. Alpha-synuclein accumulate in mitochondria. Increased alpha-synuclein in mitochondria lysate, it's also done by us. Here we use another method, to show that that alpha-synuclein is affected by, and using that GC1 cationic dye, you see a shift from the green color to the red color, which indicate also that it's affected. Neuropathology summary in this case is loss of dopaminergic neurons in substantia nigra, Lewy body in neurons and neurites, and alpha-synuclein tau accumulation increase. Implicated pathway are inflammation, ROS, autophagy, apoptosis, neurotrophic factor. In the PD, so Parkinson's disease is multisystemic disease. Then the conclusion will be this. The alpha-synuclein is the most enriched microRNA, associated with myosin 5A. This is based on the paper which recently I read, but we did not have the data yet, but I believe it is really associated with myosin 5A alpha-synuclein in my model. The next one was genetic analysis we discussed yesterday, and so I'm going to go over very briefly. What I said yesterday, for those of you who were not present, was the fact that the chromosome 8 is located, myosin 5A, and in our case, it's the exon 4 affected by a point mutation of this myosin 5A. And we did a study in a control, and also affected homozygous rats. And this is the showing exactly how those things happen. Yesterday, I showed exactly the same thing. We look at the myosin 5A in affected one is not present. It's present in the BD4, and it's present also in the Sprague-Dawley original rat. And the myosin 5A mutation we mentioned yesterday that affect humans in Griscelli syndrome, affect horses in Lavender syndrome, and affect mutated mice, and also it's a mutated rat, reported in Japan, and this model too. Well, we have some question here, we need to solve, we need to think about, myosin 5A interact with alpha-synuclein at the presynaptic terminals, have to be determined. Myosin 5A influence accumulation and or aggregation of alpha-synuclein in the presynaptic animal. Is myosin 5A play a role in PD and Alzheimer's disease? Remains to be determined. The future direction is to explore genetic alteration responsible for disease, explore the role of DOPA, we discussed yesterday, because the metabolism of DOPA is also altered in this model. Also identify protein for developing disease-modifying therapy, explore the potential involvement of myosin 5A and genetic alteration in humans. Here is what probably we need to talk about when we have discussion. We don't have time, no? The time is over or not? I still have time? Okay, because yesterday in our talk with Dr. Tutsi and Dr. Bruno, we discussed about this type of circuitry involving Parkinson's disease, the direct, indirect pathways. And I said, well, it's good to present a kind of a figure like, to see how does it mean, And you can see here on this figure here, you can see very high schematic representation of head of a rat. It's a sagittal sections. And you can see the connection. The substantia nigra is here. You see the substantia nigra is here. And you see the substantia nigra is sending projection here in different parts of the brain, including thalamus. The thalamus is also, because of the stimulus, is projecting into the cortex over there. Then you see here the direct pathway, which connect the striatum with the substantia nigra, or the substantia nigra connect with the striatum here. And it passed through to the GP internal part, but is not passing to the subthalamic nucleus here. That's the indirect part over here. And so the connection here is any time when you see here from the cortex, you have a lot of glutamatergic neurons, pumping up glutamatergic excitatory stimulus to the striatum. The striatum is very huge part of the brain over there, and it's very significant, because the dopa is coming over there like a moderator, to try to keep pace, to maintain the normal homeostatic environment in the brain. If the dopa is not there, you have alteration in many, many sections. What did bombardment of the glutamate, coming from the cortex into the striatum over there happen? If you don't have the dopamine over there to keep them in track, to maintain the homeostatic balance over there, a lot of neurons probably will die over there directly or indirectly because of too much excitation over there. It's so-called the Chinese glutamate syndrome. You remember that story? When don't go too much to the Chinese restaurant because they contain too many glutamate over there, and that kills the neurons. And so this analogy, with too much excitation, from the excitatory transmitter from the cortex over there into the striatum can induce damage over there. Then you see the indirect pathway over there. It's also involving the basal ganglia with many other nuclei. And when you think about what happened in the brain over there, as I mentioned, as mentioned by many others in my yesterday and today, that is interconnection with many other systems here. You touch one system, you trigger a chain reaction in many other places of the brain. And so gangliosides, they also can be this problem. You have a problem in some nuclei over there, but they are connected with other centers, and you see a response from that part, Now, the next one, the last one to synthesize here, because also we discussed yesterday, about prion-like communication. What I believe, probably I need to update this figure here, is that yes, might be true, might be not true, but the prion, is an infectious disease. This one is not an infectious disease, one part. The other thing is like this one, it's possible, that they accumulate alpha-synuclein from two reasons, In my case, you have plenty of astrocytes and microglial cells there. You have so many factors here, which can induce degeneration of the neurons. They release the alpha-synuclein outside. The alpha-synuclein can be uptaken by the healthy neurons, and accumulate here. And when accumulate here, they try to go here at the presynaptic site to communicate, send the message on the opposite neurons over there, postsynaptic. But myosin 5 is not present over there, or not functional over there. They accumulate over there, and when they accumulate over there, they force so-called loose body life. And the neuron die. And the process continues in this way. Die, and exocytosis, endocytosis, going to the next neurons over there by endocytosis. It's possible also to communicate from neuron to neurons, like it was in prion theory, But I still have some doubts about that happened in Parkinson's disease. So with this in mind, let's go to the final stage. Parkinson's disease pathogenic mechanism, a lot of factors. Forget about, I already talked about this one. There's no time. Here is Michael Fox. Michael Fox, I have to thank him very much, because at the time when 2012, I believe he came at a meeting in New York Academy of Science, keep a speech over there, and he said, guys, I would like you to solve the problem, and be careful because it's not coming from the sky. You have to ride or walk up on the ladder over there and pick up from the sky and find out the solution to solve the problem, because I am and a lot of people are diseased with this problem, And so I thank you very much, and that's all I have for today. The time is over.